Samir started his clinical pharmacology career after obtaining a PharmD and serving as a clinical pharmacist in a research hospital where he started a therapeutic drug monitoring service using Bayesian feedback. His research focused on developing population pharmacokinetic and pharmacodynamic models to help clinicians select the best dose for the patient with a focus on pediatrics. In 2007, he joined Farsight Consulting Services and helped sponsors to apply model-based drug development for more than 100 studies. These contributions generated more than 60 abstracts and manuscripts in the field of pharmacometrics, spanning numerous therapeutic areas. Dr. Mouksassi earned his PhD in pharmaceutical sciences from the University of Montreal Faculty of Pharmacy. He is also a fellow of the American College of Clinical Pharmacology. Samir earned his PharmD from the Lebanese University in 2003. I'll now turn it over to Dr. Mouksassi to begin today's presentation.
Hello everyone and welcome. Today's topic will be on powering pediatric studies, a seamless approach for meeting FDA's pharmacometrics criteria using Phoenix NLME and trial simulator. And I will be pleased to share with you our experience on how we help numerous sponsors and clients to meet regulatory requirements with focus on FDA's criteria. First, a brief introduction into the challenges and opportunities that we typically face within pediatric drug development. First, we have a tremendous clinical need, and the challenge is we want to be able to find a safe and effective dose in this very vulnerable population. A dose that is easy to administer and easy to compute, because most of the time we can just give one dose that fits everyone in kids. The problem is the current status of the practice is highly empirical. Most drugs are used off-label, and many times we've seen doses that are linearly extrapolated into this population, i.e. using a kind of simple milligram per kilogram or milligram per square meter extrapolation that might not be appropriate all the time. In this specific population, we have a lot of difficulty of collecting data. We have difficulty in collecting, for example, pharmacokinetic sampling because of a small kid. We can't collect a lot of blood. We have a challenge of PD evaluation because small kids we can't just ask how they feel or we can't conduct typical evaluations that are feasible in adults. Also, we face a lot of ethical issues that vary a lot between different regions. And most of the time, because of it is very hard to collect data, we need to run in multinational studies to be able to collect enough patient and information, and we need to face this different ethical issue between different regions. Also, many times, the PK-PD relationship in this specific population is not known. But we have a lot of opportunities that will help us remedy all of these challenges. First, most of the time when we reach pediatric drug development, we already have a lot of prior knowledge, whether it's from adult data or prior off-label use, and a lot of disease knowledge as well. Knowledge about pharmacology, PK, and possibly PD. Other incentives to sponsor are that if they get a pediatric indication, they can extend the patent life. And this is part of several regulatory initiatives that are meant to encourage sponsors to invest into pediatric drug development. Namely, we have the PREA, BPCA, and others. And today I will show you how using quantitative tools will help us understand the most of the data in this limited population. So what leads to pediatric drug development? Basically, what we've seen most of the time is sometimes sponsors get an FDA written request, or they are preparing their Europe submission where they now need to provide a pediatric investigation plan as early as phase 1 in their drug development program. In any case, they will need to provide a justification and rationale to run a pediatric protocol. And here I got an arrow that shows that sometimes we have some discussion between FDA and EMA, so it's not uncommon to see like joint requests of the two agencies into running a pediatric protocol. Mind you, it's not all the times that we have the same request. Sometimes what FDA asks is slightly different from what Europe asks. But today, our main focus will be on our experience with FDA. So I wanted to share with you a sample of a written request by FDA. So typically, a sponsor seek our advice on how to handle this special request. So here, that's a written request, and in most of the time, the written request will ask to characterize the pharmacokinetic profile following multiple doses in specific pediatric age group. And why that's we want to be able to compare with adult PK data to identify the dose that is appropriate into the different age group. And that's needed to be able to bridge plasma concentration between children and adults. Sometimes PK-PD relationships are not known, and other measures need to be taken into account. In any case, FDA always encourages the sponsor to maximize the trial success. We need to run some kind of clinical trial simulations, and that's what I will be showing later in this presentation. Other items that need to be addressed within a FDA written request, very important, is blood sampling. So how many samples I need, how many subjects I need, and the goal of the design will be to have a design that ensures precise estimation of primary PK parameter. In this particular example, FDA has asked of a design that allow us a good precision on clearance and volume of distribution, the central volume of distribution. So here it's mentioned that we need a good design, precise estimate, but what is a precise estimate? What is a good enough precision? And we got this publication from FDA that clearly clarified and specified what is meant by precision criteria to be met before proposing to run a pediatric protocol. And namely, the design should have as an endpoint to have more than 80% chance of success or power to meet the precision criteria of having 95% confidence interval of the geometric mean between 60 and 140%. And I will tackle this in more details briefly. Just a note here is, again, in this publication, given the complex nature of nonlinear mixed effects modeling, clinical trial simulation is generally needed to derive the appropriate sampling schedule and the sample size. So again, given all the challenges we have, the best way to address all of the assumptions is to conduct clinical trial simulations. So basically, from this paper as well, they give an example where, imagine we have a PK model with a clearance equation that is allometrically scaled by body weight, and possibly we have a kind of a maturation function of age that take into accounting possible developmental changes. So here we have an equation that jointly have body weight and age into the same equation. And we want to predict what happens in various age groups. For example, here from the written request I just showed you, we got these age groups to address. And it's important to know that sometimes we don't need as much resolution into age group. So the division into age group might be dependent on the drug we are dealing with, because sometimes we can just, for example, have below 12, above 12, from 2 to 12, 12 to 16, and so on. So our problem here is typically we have an estimate for theta here and standard error. Also, we have an estimate for the Hill parameter and standard error, but we want to compute what is the clearance in, for example, 6 months old. So to be able to do so, we plug in age, but we need to find what weight to use. And we need to find the weights that correspond to 6 months. And second challenge will be how to compute the precision on this equation, which involved several parameters with several standard error on each parameter. And later on, I will show you how we can do that. So to sum up, after a written request, several items need to be discussed with the health authority. Basically, what dose to use? Are we using a fixed or a milligram per kilogram dose? Are we cutting by weight groups or by age groups? What is the number of subjects and number of samples needed to meet the precision criteria? What analysis method will be using? Most of the time in pediatric population, we encourage to use population PK analysis because of the sparse nature of the data and because it's very hard to collect enough data to conduct non-compartmental analysis. Also, many times we combine prior adult or other data with the newly collected data to increase our chance to determine the PK parameters with enough precision. And sometimes we propose a kind of sequential analysis of patient. So, for example, we run a protocol with n equal 10 patient. We see if we reach the criteria. If not, we continue enrollment until we reach the criteria. And also, within the drug development program, it's very common that new data is generated which warrant frequent amendments of the possible pediatric protocol. So a pediatric protocol can be a dynamic document where it's frequently updated according to the latest development on what we know on the PK, PK-PD, and PK efficacy. Today I'll be covering a protocol that involves PK only. Sometimes it's not enough. We need to conduct a protocol with PK-PD or more complex with PK efficacy. So what I will be showing you is how the key methodology to answer all of these questions is a combination of clinical trial simulation combined with estimation and refitting. Easier said than performed, there is many challenges to this framework. So first, we need a model in place, so a PK or a PK-PD model that we build on prior adult data, for example. We need to customize this model to incorporate some scaling component, potentially a maturational developmental changes component. Next, as I showed you into my clearance equation, we have weight and age. So we need to feed into this equation a realistic covariate distribution. And to do so, we have developed a model that would allow us to generate a realistic age-matched distribution of body weight. So we can, with this model, we can simulate, for example, at any age group, a realistic distribution, and more details will be given shortly. Next, once we have a model in place, a realistic distribution of covariates, we want to know what is the minimum number of patients, number of samples to have precise PK parameter. And to do so, we use D-optimal or ED-optimal design software like PEFIM and others to give us an initial study design. But the optimal design given by this software assume perfect execution scenario. And that's why after we have an initial trial design, we feed everything into trial simulator, which is a very flexible and powerful software that will allow us to test not a perfect scenario trial, but a real scenario trial where we can incorporate dropout, BLQs, missed samples, and many other possible protocol deviations. So we simulate the trial and we refit it. And maybe this initial design is not successful. So we need to do several iterations of this framework, basically the simulation refitting, until we reach a good enough trial, which is a trial that would give us 80 to 90% chance to meet the criteria. So here I will cover the various software and technologies that we use in order to perform this simulation refitting framework. First, I will mention our trial simulator software, where it is specifically designed for protocol simulation, for clinical trial simulation. It has a very user-friendly interface that helps in coding simple and complex trial designs. It supports protocol deviations like missed samples, BLQ, even time deviation of sample collection, and many more. It can read a covariate file as input, and it can generate fitting-ready datasets. So we run trial simulator, we generate fitting-ready datasets that we can later on directly fit. And it has an interface that is very flexible to change the design. Namely, if I run iteration 1 with number of subjects that were not successful, I can very easily change to a higher number of subjects or change the collection and samples. And I will be demoing these features shortly. Also, it allows us to add uncertainty and other levels of random events. And just a mention here, it's very, very hard to code protocol design and deviation into other software tools. And again, I will be demoing this feature maybe in a few minutes. So once we simulated the protocol, we want a solution where we fit the simulated data. And our tool of choice is Phoenix NLME for several reasons. Basically, first, it has a user-friendly interface that allows us to fit models by replicate. So here, to be able to compute power, we need to repeat the trial maybe 200 times or 1,000 times. And on each replicate, we want to fit the replicate and data and see the outcome. So the sorting functionality allows us to use one model sorting by replicate to get our answer. Also, it has a very convenient secondary parameter functionality to compute the relative standard error percent at specific weight and age. Basically, I just need to include one more line of code to get, for example, what is the clearance at 5 kilogram. I don't need to add any other code. I don't need to export a third-party software. So, for example, in the other software, I will need to be able to refit each replicate. It will require significant coding programming efforts. I will need to separate the simulated data into separate files and or generate the number of replicate of control files. So if I have 200 replicate, I will need to generate 200 control files that read the appropriate replicate data. I will also need to use R code to compute secondary parameter standard error using the delta method. But all of this is directly incorporated, and it's an out-of-the-box feature of this software. As I mentioned previously, simulation refitting may require several iterations, and that increase the burden of programming. Imagine that every time we need to reevaluate a design, for example, I want to increase the number of samples, other softwares, we need to regenerate data template. So it's really more time consuming than just using a one box, one value to change into trial simulator. Sometimes we need to take into consideration as well that some designs are not feasible in the clinic. We need to take into account the nurse times and so on. So all of this will require several iterations until an acceptable and final design. So overall, that's the framework after I explained a little bit more detail about each component. And so first, we need to modify the prior model. So first step will be to customize a previously developed adult model so I can use it into simulation. And we might have in-house data from the adult development program. Sometimes it's only using literature data, a published model in adults, and another company is basically developing a pediatric indication. So in the most simple of cases, imagine that we have a model, a clearance equation that we want to incorporate some kind of allometric scaling into it. So we refit the model including this allometric scaling. So again, as I mentioned, we use Phoenix NLME because it has a user-friendly interface and it allows fast customization of the models and to very easily add allometric or maturational functions. And so after we customize the model, we want to know what is the dose to use in the various age group. So we can do so using some kind of simulation. So here I'm using a graphic from one of our publications to show that for this particular application, we found the different doses to use in the various age group via simulation. So here, for example, from 6 to 12 months, we are using 0.05 milligram per kilogram per day. For 3 to 6, we are using slightly lower dose. And in the very young, we have a narrower split because we could not find one dose that will allow us to reach 90% of target from 0 to 3. So So we had to split it from 0 to 1, 1 to 2, 2 to 3. So all of this is to say that we need to find the correct dose in the various age group. And here our criteria was we want to find a dose that allow us to have at least 90% of pediatric patients within the target window. And here the target window was the range of exposure seen in adults. So once we know, we customize the model, we know what dose we used, we want to plug in the realistic distribution of Covariate, and we have developed a model based on CDC data. We all know, and the growth charts, basically this is available, frequently used, and these growth charts are based on some statistical distribution models. So here we have some kind of smooth distribution of body weight versus age, and we can use this data and this model to conduct simulation. So, and in the same paper, we have developed and applied this methodology where we downloaded the data, we developed a flexible model, a gamma LSS model that will allow to simulate. So here we can see that we can simulate normal population, or most frequently we can have a very special population. For example, a low body weight population or an obese population. So we need to be able to manipulate this distribution and to select, for example, below normal weight or above normal and so on. And for example, using this model, we can simulate normal, low body weight, malnutrition patient, or obese. It's very, very flexible. It's a statistical mathematical model that we developed to allow us to run in various populations. So the realistic Covariate distribution, again, can be based on in-house data, off-label data, or literature data. And it's very easy to be customized, to customize the normal population model into a disease-specific population. So let me now go to the live demo to show you in more detail how all of these different tools are bridged. So I'm sharing here my desktop, and I believe you should be able to see my Phoenix desktop here. So basically the first step, I have a model where I have an adult model, and here I'm showing you the interface of a Phoenix population PK model. It's very flexible. So I have a visual depiction of the model. I have some parameters. It's very easy to, for example, here I'm already using body weight as a covariate on volume and clearance. Imagine I don't want to use an estimated power. So I can just plug in the value I need. So here it's already 1 or 0.75. You can see the previous value that was estimated was 0.56. It's sufficient to just type in the value and fix it, and I maybe have a model that is suitable to pediatric simulation. But maybe I want to modify the equation. So here in my equation I have body weight, I have also creatine clearance. But in some cases I want maybe an additive creatinine clearance. So it's very, very easy to switch to edit mode and modify my equation into maybe an additive creatinine clearance effect. Maybe I don't want a power model, I want just a linear model. So it's very, very easy to edit and modify. And I refit my model. So here, basically, I got 4 different models, one with theoretical allometric scaling, one with an additive renal function, and one with a multiplicative renal function. So here, if I open this model, you can see that I have this one where I'm fixing the volume to 1 and clearance to 0.75. So it's very flexible and easy to customize a previously developed model. It's also very convenient to compare the various models that I want to test, final versus theoretical. I can compare goodness of fit and so on. So first step, as I mentioned, is to customize the adult model. Next step is to generate a realistic distribution of covariate. And we can do so through Phoenix Connect. Basically, with Phoenix Connect, we can run various third-party software from within the Phoenix platform. And this is very convenient to keep everything within a workflow. So we can run non-mem, SAS, sigma plot, S+, PSN, and R. And here I'm using an R script that codes the gamma LSS model. Basically, I can just tell the script I want to simulate from what age to what age, and everything is automatic. So once I specify what age group I want, it will generate a bunch of graphs. So here it's simulating from age 2 to 17, boys and girls, and it's providing various quantiles, and also it will give me the data that I'll be reading into trial simulator. So here I have simulated a age-matched weight from 2 to 17. I can control the age limit and everything else. So after I generated the covariate distribution model, I can code my model into trial simulator. So that's the same model that I showed you in Phoenix, coded here. And as I showed you as well, we had 4 different models for the clearance. So here, I'm using 4 different equations, 4 possible equations for clearance. Basically, some maturation, additive versus multiplicative type of model, and so on. Just an example to show you how flexible is to use this software, and with the possibility to have as well multiple possible models. So here I've coded the model. You type in your parameters values. What's very interesting into trial simulator is that we can very easily control the enrollment. So I'm starting with total number of subjects in study equal 20. So, and I will show you how we can modify this very easily. I can have different treatment arms. So here there is 0.06 milligram per kilogram dose. I can automatically scale by body weight, depending on the treatment arm, but what I really want to simulate is I want this dose to only apply maybe above 6. So in assignment, I can define age strata. So here I'm using as an example 6 years old. So I can have less than 6 and greater than 6 age. And then I can tell it, give treatment arm 1 to only for patients that are less than 6 years old. And it will automatically assign an equal number and so on. It's very, very flexible. And in simulation scenarios, basically, here is the power of running multiple possible scenarios where I can tell it, okay, I want which different model I want to use, model 1, 2, 3, 4. Here I'm just selecting model 1 and 3, for example, to compare theoretical scaling versus empirical. I can as well easily change the number of subjects. So I can just type in maybe the 30, increase the number of subjects, and so on. In observation, what we typically do is we input whatever the optimal design software has given us as optimal to determine the PK, with the possibility to include some time deviations as well. So once we simulated in trial simulator, we export the output and we import it into Phoenix. So here I have data that was imported directly from trial simulator. And what's very nice, suppose I rerun my simulation, there's a feature here that allows us to refresh from source. So if I changed or I updated my trial simulator output, I can just refresh from source. It will reread the CSV files that I generated, and everything is linked through a workflow. So here I do some kind of data management, and I'm refitting. Here I double-clicked my model. It's opening. And that's a huge data set. So what I wanted to show you here is I can sort by population or by replicate. So I refit each replicate by sorting by the replicate, and what I will get is I will get parameters for each replicate. So here for replicate 1, 2 replicate 200, I will get estimate for clearance, the CV % of clearance, and 95% confidence interval. So, for example, I can then see if this particular replicate was successful or not and compute the power by doing some extra data management, again, within the Phoenix platform. Everything is integrated and linked to the same platform. So here I'm generating an HTML table that can directly go to a report. I will go back to my slide deck. So basically, I've shown you how we can integrate the R covariate simulation into the trial simulator, and everything linked under the Phoenix platform. Here, I showed you that we are using the optimal design as a starting point. It involves some assumptions, namely perfect trial execution, and within the clinical trial simulation, which is the preferred methodology, we can take into account missing sample, realistic distribution of covariate uncertainty, so we can compute the power with respect to the actual trial execution and outcome. And it's very, very easy to incorporate and test alternative scenarios, different number of subjects, different number of samples. I can easily modify the dosing schedule. I can easily assign a dosing schedule specific to an age group. So what I didn't cover, I didn't show you into like the visual demo, is that I input the adult allometrically scaled model into optimal design software to find the starting number of subjects and starting number of samples and time of samples. And the only drawback of this software, well, it doesn't incorporate any trial or protocol deviation execution. So that's why the gold standard will always be to conduct the trial simulation. And once I run my simulation, my replicates, I want to summarize it and incorporate the results into a table so that I showed you a table can be generated within Phoenix. What I didn't show you, we also have what we call Phoenix Connect Reporter. So I will share my desktop again, show you another example. So here, any object, any table you have in your workflow, you can include a reporter object. You can select, you can add content. For example, here I have some R-generated graphs. I can tell it, okay, I want these 4 figures to be incorporated. I selected these figures twice. I'll show you once I select what content I need or I want, I can just run it. I did. And it will generate a Word report with whatever content, figures and tables I chose to include, which is very convenient and saves us a lot of time. I double-click the Word document, and these are generated graphs which were incorporated automatically into the Word document. You can also control the captions, the titles, and so on. So that's one nice feature. Going back to my slide deck. So basically that was a table generated within Phoenix Connect and our RTF tools and automatically output into a Word document. So, in summary, to be able to simulate an actual trial outcome, we are using different tools under the Phoenix platform. Basically, Trial Simulator, Phoenix NLME, and bridging everything with Phoenix Connect. Just of note here is future versions of Trial Simulator will be under the Phoenix platform. Currently it's not, but future version will be for better integration and refreshability. Before we wrap up, just a few notes on why we need sufficient precision. Why is the focus on this sufficient precision for pediatric trials? That's because once PK parameters are characterized with sufficient precision, within each age group, simulations can be performed to find the best dosing schedule by age group. So sometimes we chose a dose, we run the trial, but the outcome was not as we expected. Fine. We don't need to rerun the trial with the alternative dosing regimen. We can just simulate, find the best dose, and we will be good to go. So here I'm mentioning a very important publication from the FDA group entitled Impact of Pharmacometric Analysis on New Drug Approvals and Labeling Decisions, which showed that and mentioned several examples on some dosing regimens, some dosing regimens that went into the label based on simulation. So it was based on pharmacometric analysis. So here I have some, and there is a nice paragraph that specifically mentions that it's not necessary that the label should include the exact dosing that was studied in the trial. Such a mindset leads to increased drug development costs with very little advantage gained from answering one question at a time. Pharmacometric approaches have allowed the FDA industry to gain deeper insights to how to best use a drug for labeling purposes, and they cite clevidipine as one example where dosage administration section was updated on the basis of clinical trial simulations. So, just a few words. I didn't cover. Okay, I showed you that we simulated the trial outcome. We simulated a protocol that has 80 to 90% chance of success, and once we have collected the actual protocol data, we fit everything simultaneously. Very convenient to estimate all the standard error, whether it's via secondary parameter or bootstrap. And I already mentioned sometimes we incorporate a kind of adaptive design where enrollment is to continue until the precision criteria is met. And within the platform and framework we developed, it's very easy to refresh all the analysis because everything is linked. So as soon as new data becomes available, we just refresh from source data and update everything by refreshing the whole project. I have few screenshots from an actual protocol data analysis. So here I have the secondary parameter output where I will get age clearance for age group 1, 2, 3, 4, 5 with the estimate, with the relative standard error %. And here, in this particular example, based on asymptotic standard error, the volume had 21%. So it didn't quite meet the criteria, but when we reanalyzed using Bootstrap, so we did a Bootstrap to recompute the standard error based on the gold standard method, which is a Bootstrap. The relative standard error was lower. So we were able to show that this protocol met the criteria using the Bootstrap. We presented the results. So here we have the 0.6 to 1.4 criteria. We present the geometric mean by age group, and we want this distribution, 95% confidence interval, to be within 0.6 to 1.4. And we showed the FDA that all the age group met the criteria. And after this work, the actual FDA response was quite rewarding. Basically, the letter of approval from FDA that we have fulfilled the pediatric study requirement for ages 2 to 17 for this application. Therefore, no additional studies are needed in this pediatric group. Also, and as I showed you from FDA publication and with some application with our sponsors, many times we have seen labels involving those based on simulation. So that's an excerpt from an actual label that we contributed to where it is mentioned that dosing simulation from pharmacokinetic data in infant and neonates suggests dose reduction XYZ. So let's show you that more and more we are seeing labels with dosing based on simulations. Overall, I have presented you with our experience and the methodology that we are currently using with the hope to meet some of the goals of pediatric drug development, namely faster trials, reduced sample size, and to be able to find the best dosing regimen for the specific age group. Modeling and simulation tools are now pivotal in bridging the information between prior knowledge and newly generated data. Pediatric data is hard to obtain, and good planning on what data to collect is essential. Thanks, everyone. And I would like now, just before finishing, to mention and to thank our many clients and partners, the health authorities for very fruitful interactions, also my Certara Consulting Services colleagues and co-authors, namely J.F. Marier and Professor Alexander Winks, my colleagues Nastya, Natalie, Leon, and Thomas, and many others,